Okay, so are we live now?
Yes. Hi.
All right.
Okay, so welcome once again. First, welcome to the audience for joining us at this plenary lecture. A brief notice that you should fill out the CMA credit form on the website for those who are interested. And we already have the extended Q&A available on the website from the talks before. And with this, let me introduce your attention to our last and plenary speaker for today. We're privileged to have Professor Thilo van Eimeren from University of Köln joining us today. He is a professor at the medical faculty of the University of Cologne and a consultant neurologist at the Department of Neurology at the same university. He is the author and co-author of more than 100 peer-reviewed publications, board member of International Study Group, and a group leader of his neuroimaging lab in Cologne. And in his work, he has invested in combining molecular and functional imaging methods in various neurodegenerative diseases. And in his plenary talk, he will address novel insights provided by brain imaging in the pathophysiology of neurodegenerative processes. And then I invite you, Professor van Eimeren, to share the screen and we can begin with this plenary talk.
Sorry, my PowerPoint just crashed, so I'll probably just share it again.
So while we wait, just notice to the audience, there is no discussion planned, but if you have any burning question, you can post them into the Q&A session and we will forward them to Professor van Eimeren to answer later on. Now the floor is yours.
Thank you, Matej. So I'm really thrilled to be invited to this beautiful conference. I followed the presentations of this last session. They were all great and very informative and interesting. So I'm really humbled to be asked to give an overview on the topic, sort of. And that's what I'd like to do. I'd like to invite you to go with me on this kind of exploratory trip where we try to find a bird's eye view on neurodegeneration and really try to find meaningful ways where imaging can unravel really the mysteries around neurodegeneration that prevent us from developing effective therapies. And effective therapies are really much needed, as you know, in the field of neurodegeneration, because it's expected that these age-related diseases will more than double within the next 30 years. So in 2050, we will have probably more than 1,000 million people on this planet who are over 70 years old. And what's more important is I will be one of them. So you have to find the cure for Alzheimer's disease, Parkinson's disease, or we need to find it soon. With cure, we could also mean that what we'd like to fulfill is that we can somehow protect the brain integrity or slow down the disease progression to a meaningful extent, so that this boundary to disease onset, the critical boundary, is, if crossed at all, at a later stage or later life stage. And what I'd also like to convince you of is that the two main forms, Alzheimer's disease and Parkinson's disease, are really more similar than you might think at first glance. And because, of course, they're both highly prevalent, they're age-related, as we said, the causes are known and all of that's true. And we have limited and very limited efficiency of therapies at this point. But both of them also occur with pathological aggregation of proteins. And we know that the pathology present in the brain precedes the clinical presentation, often by decades even. And there's this hypothesis for both of these diseases, as you heard by all the presenters today, that the pathology seems to spread within brain networks. So this is something that both of these diseases shares. And as you heard from Matej as well, there are overlapping forms such as dementia with Lewy bodies. And the history of these two neuropsychiatric diseases is really also a history of discoveries made by imaging, and this is mostly optical imaging, and it really starts with the light microscope on the left side, and this is the actual microscope that Alois Alzheimer used to describe the groundbreaking discoveries of the cellular bodies. That was back in 1906. The cellular bodies like the plaques and the tangles that are associated with the disease then named after him. But Friedrich Levy also found these bodies in dementia with Lewy bodies, but later it's been known that these cellular bodies are also present in Parkinson's disease. And also Arnold Pick described cellular bodies known as the Pick bodies in Pick's disease, frontotemporal lobar degeneration. And then only with the advent of new technology, which was the electron microscope, we were able to zoom in to those bodies and identify that they were made up of aggregates of protein foldings. And we identified, or others identified, of course, that the tangles were made up mostly of tau as a protein. The Lewy bodies, the specific component is alpha-synuclein, of course, and the plaques are made up of beta amyloid structures. And now, with the advent of cryo-electron microscope, we can certainly have a very optimistic view of the future, that we will be able to really pinpoint to the atomer level these protein aggregates, and this will help us in designing drugs and but also molecular imaging markers for these kind of diseases. These protein pathologies in these diseases have also a very interesting commonality, has been referenced by most of the presenters today, and that's that from a certain point of origin, that's debatable, there seems to be a spatial spread. So with the years and with time, there seems to be a more or less prototypical spread of that pathology across brain sites. And this spread of pathology seems to be quite specific for the type of protein involved. And it's been debated since then, or the hot debate around this is that kind of spread, is that a cell autonomous function or type of initiation of the pathology, meaning that it's different vulnerability factors within these regions that promote the pathology. Factors like genetic expression profile, but also the wear and tear of activity can play a role, or other types of vulnerability factors might actually govern the occurrence of these pathologies in the different stages of the disease. And the other or contradictory position would be, no, it's actually like a cell-to-cell transmission. It's a non-cell autonomous induction of misfolding by cell-to-cell transmission, like a prion type of spreading, like Nikolai said. It could be the case. But we have to bear in mind that in the end, what this is only telling us, when we look at the post-mortem information where in the brain these type of protein aggregates are presented, it's a little bit like looking at the still of a famous movie and trying to infer the whole story that led up to that point. It's impossible to do. So we need in vivo information. We really need some kind of a story. Oops, sorry. So I hope it's not going to crash all the time now. I'll just try to pick up where we left. I think it's something to do with the simultaneous thing of the two. So I'll try to catch up. All right. So we need in vivo information, and this is something that we can hope to get from different types of imaging, molecular and structural imaging. But at the same time, when we broaden the scope again, we think that this type of pathology develops really at a crossroad much earlier than that, and a crossroad of maybe innate biological factors such as genetics, certain lifestyle choices such as the amount of sleep that you get, the amount of exercise that you have, the amount of education that you received might play a role. But also external factors might play a role. And we just learned by the COVID pandemic how easy it seems to be for certain viruses to enter the brain. So viruses can certainly play a role in the initiation of pathology, but also neurotoxins play a role. And the real problem with the protein pathology is that we don't really understand the pathophysiological cascade. So we don't know if protein aggregates are really a causal substrate, or if they're just an epiphenomenon, a sign of the dysregulation of cellular processes, or even in the form of aggregates, could even be a protective mechanism of the cells. So again, I think that brain imaging is the perfect in vivo window into these dynamic processes. We have heard about metabolism with FDG PET. We have ligands now for many of the protein aggregates. We have ligands for neurotransmitters. We can explore that. And of course, we have the power of high resolution MRI with functional and structural brain networks. So let's start with the Alzheimer's disease, and you've heard much about it, so I can just skip that. So it's tangles and plaques. Tau tangles and beta amyloid plaques that play a role there. And the general idea of recent years, also promoted by Clifford Jack and other illustrious people, is that that there is a cascade of pathology, meaning like domino pieces, one has to occur and fall so that the other pieces basically fall. So the first thing that probably needs to occur is amyloid, beta amyloid pathology. And then at a later point, for reasons that we don't really know, tau pathology seems to occur and spread across the brain. And with that, neural injury occurs and structural decline. And then, of course, at a later point, cognitive and clinical decline. So with the means that we have in Cologne, for example, we can really measure now the amyloid pathology, we can measure tau pathology, we can measure FDG hypometabolism and the structural phenomena that go with that. And one of the first realizations we had with that was a study that Dr. Bischoff did in our lab, looking at amyloid, FDG, and tau. And for the first time then, we realized that there is a very close spatial and temporal relationship between FDG and tau. Now that's old news, but at the time it was quite substantial news. that not amyloid is related to FTG hypermetabolism, but tau seems to be. Tau in the spatial extent, and we heard also from Nikolai, is really an important predictor of the clinical progression, much more than amyloid seems to be. And this is just one of the early examples where tau pathology is clearly better than MRI pathology to predict progression in Alzheimer's disease. We have recent data in our group by Julia Pfeil, who looked at the regional distribution of amyloid, that also the regional deposition of amyloid plays a role, or can play a role, for the progression, for the clinical progression to AD. And we now also try to use, with Elena Döring's work, the mighty power, almighty power of machine learning to predict clinical progression by baseline data, such as the clinical disease state and ApoE genotyping, for example, and FTG, and that's going a long way, as it seems. But we know that these are important biomarkers, but markers are not makers. So we need to understand how that progression, or how that protein pathology really instantiates and propagates across the brain. And very important work has been pioneered by Mark Diamond and his group in Texas, but also elsewhere, where human protein strains have been taken from deceased patients and then purified and amplified, so that you have a certain number of monomers of, for example, TDP43 or 3-ART tau from Pick's disease, for example. And then this is injected into a rodent model. And what they observed is that after a time, really a very specific set of brain areas seems to be affected. And these brain areas resemble quite well the brain areas that are affected by the protein aggregation in the human condition. So this speaks to this idea that there really seems to be like a protein-like, a prion-like propagation of the disease. At least there's a propagation of the protein across the brain, and the misformation and aggregation. And therefore, I think it's very important work to look at these different tau strains very carefully, and this has been done by cryo-electron microscopy. This is just an example of important work from Cambridge and Michel Göder's group, and these beautiful images of the tau fold in Alzheimer's disease, in Pick's disease, and also in chronic traumatic encephalopathy. At the same time, and we heard this already today, even with the presumably very similar tau strain in Alzheimer's disease, we get different kind of patterns, and this is related to the clinical pattern, and for example, frontal involvement, or the leukopenic variant, or and posterior cortical atrophy as very rare condition in Alzheimer's disease. And this is related to presentations of tau in these areas. So the tau strain doesn't really necessarily govern exactly where the tau is going, and and this is just further evidence that even within the domain of the typical AD, there seem to be different types of trajectories discernible, and this leads to or led us to the original idea to look at the tau PET and dissect them in the independent components analysis. This is very similar to what you heard from Nikolai, but much earlier work there. Just to summarize that again, so you have this with resting state fMRI, you can dissect those in the main components of connectivity, and you can do kind of the same trick, and Dr. Merle-Hönig did that with the tau PETs and thereby discern the tau pathology networks that are most present in a cohort. And then when you look at these networks, sorry, go back again, they look very similar to the resting state networks that you get with resting state fMRI. And what Melle did at the time, she also looked at the most expressed or highest peak of tau pathology within these kind of networks that would resemble the terminology epiclusters that Nikolai used. And if you use these as a seed in a resting state connectivity analysis, you get more or less the same kind of networks that resemble, again, the Stanford networks of resting state connectivity. So again, it seems that all of this is pointing towards the importance of the connectome also governing the propagation of the disease. And if you take this all together and we summarize this, Dr. Bischoff and others in this publication, you get like a triad of factors that rule the temporal spatial pattern of a neuronal dysfunction. And that is the protein strain, of course, but also the connectome and also cellular features such as the presentation or the amount of protein that is present in these cells. And imaging, functional, molecular, structural imaging is really the perfect tool to bring all that together and to investigate that further, to be able to predict the propagation of tau. And this is something that we currently do in a larger longitudinal study by the scientific lead of Dr. Bischoff, looking at different factors that govern the propagation over time, trying to assess then the factors and to predict in a precision-based kind of precision medicine approach what the future might hold for these patients. So now switching to the other topic, the much older topic, Parkinson's disease. 200 years ago, more than 200 years ago, James Parkinson's description of the disease is still very much alive, and then I already mentioned Friedrich Levy, who more than 100 years ago described the cellular bodies. But 50 years ago, more or less, we had these four people: Ole Honigkiewicz, Arvid Carlson, Eric Kandel, and Paul Greengard, who were all important in describing that dopamine loss or loss of dopaminergic neurons is really the pathophysiological hallmark that brings up the Parkinson's disease. And that's been very important in developing also the therapies that are so effective in Parkinson's disease. 20 years ago then, more than 20 years ago again, Maria Spilantini and Michel Bödert published this important work on alpha-synuclein as the most important ingredient in these Lewy bodies. And then 15 years ago, I already mentioned Bragg's hypothesis, or the Bragg's described this prototypical distribution or propagation of alpha-synuclein pathology from the medulla over the brainstem and to the cortical levels of alpha-synuclein pathology, Lewy body pathology. Now, this view has been supplemented in recent years by the idea that maybe the pathology, the alpha-synuclein pathology, doesn't really start in the brain, but it may start in the gut already, and then the pathology might be transported by the vagus nerve to the brain, to the medulla and the brainstem. And that might have something to do with microbiota, might have something to do with bowel inflammation and a leaky barrier. That's all unknown, that's all new hypotheses. And another new hypothesis, even newer, is the idea that maybe the spread isn't that prototypical after all. Maybe we have a clustering of the spread into one kind where the origin of the alpha-synuclein and Lewy body pathology might be in the intestinal system, and then there's a spread to the north, let's say, and then finally it ends up in the substantia nigra, where it causes destruction of the dopaminergic neurons, and then further on marches to the cortical areas. But the opposing or the other possibility could be a brain first type of spread, where the pathology starts in the limbic system, in the amygdala, for example, and then spreads southwards over the brainstem to the intestinal system. And this idea has been put forward mostly by Per Borkhammer and colleagues in Aarhus in Denmark. And they propose that what happens is with the brain, with body first PD, you get a very or more or less symmetrical spread of pathology with a symmetrical loss of dopamine in the substantia nigra and the stratum. Whereas if the brain starts in the, if the pathology starts in the brain, it starts at one side only, and then you get a very lateralized pathology with a very asymmetric fluorodopa or FP-SID signal in the stratum, and this was something that we wanted to test, and this is something that Magdalena Bannwinkler, who recently joined us from Austria, is now testing, and with PPMI data. And what she found so far is that, well, of course, there is an increased asymmetry in the putaminal dopamine transporter SPECT density in PD in comparison to healthy controls. But there's still some of the patients are not so asymmetric. So there are some of the patients who really are not so asymmetric in their extent of dopamine loss. And she also found that indeed there is amygdala atrophy in Parkinson's disease in comparison to healthy controls. But what she also showed here, or shows here, is that there is no correlation between the asymmetry in the putamen and the size of the amygdala on that side, basically on the most affected side, which would be hypothesized by the brain first hypothesis. And therefore, and also if you dissect the group by RBD presentation, or just look at the more and less affected hemisphere, there is no difference in the volume of the amygdala in Parkinson's disease. So this paper or this future paper will show no support for the brain first and body first hypothesis. Doesn't mean it's not true, it's just there's no support there. One of the reasons why that might be the case is actually that the link between alpha-synuclein and neuronal cell death isn't that clear after all. And these are a couple of slides that I got from Alberto Espay from Cincinnati, from a recent presentation that he did here in Cologne, where he showed that, for example, what you would assume, and this is a graph basically showing you the Hoehn-Yahr score on the left side, which is the very coarse clinical severity scale, and on the other axis is Bragg stages, so the pathological severity scale. And you would assume that these two kind of severity scales should correlate in a way. So the more the disease severity is present, and the more advanced the Bragg stages should be. And the reality of the data is actually quite different. So it's across the board. At the same time, you would expect that patients or subjects that die without Parkinson's disease symptoms should be without with a Bragg stage of 0 or 1 or 2, but not more than that. And the reality again is that it's all over the place, with a lot of people with stage 4, 5, and 6 of Bragg without any PD symptoms. So, and again, this is just, it's not necessarily, let's say, proof that this hypothesis isn't true, but there's little support if you really look into the data that Lewy body formation really is related to cell death. And this is another example where you would expect an increase of Lewy body presentation with duration of PD. Again, it doesn't seem to be the case in the pathology confirmed PD patients. Another example, which is maybe more important even, is that you would assume that with an increasing number of Lewy bodies present in the substantia nigra, you get a decrease of numbers of nigral neurons. And again, there is no correlation like that to be found. So that all kind of puts the question in our head if maybe the Lewy body pathology is just an epiphenomenon of Parkinson's disease, not really directly causing the cellular death, but it's associated with the disease for sure. What we know, however, is that the disease starts only when a certain number of dopaminergic neurons have already died, and the degeneration of dopaminergic neurons is really a well-established marker that correlates well with the clinical progression. So we know that this utility is high for the diagnostic purposes, so we can rule out other reasons than Parkinson's disease. That's also why this has now been mentioned in the clinical diagnostic criteria for Parkinson's disease as a rule out, a non-positive DaT SPECT. And it leads to a change in diagnostic and management in many patients, as we know. But what we now want to know is how well does that in fact correlate? And we know that the motor symptoms in Parkinson's disease can be measured with the UPDRS score, the part 3 score of that, and we can measure the dopamine integrity by using the dopamine transporter SPECT. So if you do that, you see some correlation between the UPDRS score, or the motor symptom severity, and the dopamine transporter loss. But the relationship isn't really very tight, right? So there is a lot of noise in the data, and some patients have high UPDRS scores, even though there is little dopamine transporter loss, and vice versa. And we think, or Dr. Merle-Hönig and Verena Jallas, who work on this, think is that resilience might play a role in explaining that variance. And we think that there could be a way to dissect these people into patients who have a high motor reserve and patients who have a low motor reserve. But in order to do that, we need to first, and that's work that Verena did, look at the exact clinical correlate of dopamine transport loss. In other words, which part of the signal really correlates with which motor symptom. And there we found that the putamen, as expected, correlates better with motor scores than the caudate. And within the two hemispheres, it's the less affected hemisphere that really correlates much better with the clinical symptoms, with the motor symptoms. And then also correlates better with the less affected body side. And that might be the case because of a bottom effect or ceiling effect, depending on what you want to call it. But the sensitivity seems to me much greater for the less affected side. And then within that, we knew that already tremor is not a symptom that correlates well with dopamine transporter SPECT or F-DOPA. So axial and akinetic rigid symptoms correlate much better. And now we can use that, and that's undergoing work in the lab. We can use that to find correlates of having a higher motor reserve that might be protective in a way for the disease. And maybe in the future we can find means to boost these kind of networks. And one of the studies that looks into this is the so-called Domoko study, which is funded by the German Research Foundation. And we expect to find out also many things about the reasons for the high motor reserve. So that goes back to this crossroad we talked about, and maybe also lifestyle factors. So, for example, exercise could play a role, but also education and sleep. Merle Hönig already demonstrated that education, for example, is a very important factor that can mitigate the effect of tau pathology, or in other words, patients who have a high education, Alzheimer's disease patients with a higher education, can tolerate much more tau pathology in their brains without having severe symptoms. And one of the reasons might be this subsequent work of her, that tau, or the amount of hypometabolism, is reduced in patients with high education. So the turnover from tau pathology to FDG hypometabolism is mitigated by education, it seems. So it's unclear what's really the driver there, but certainly education is not helpful in that regard, not harmful, I would wanted to say, in that regard. So what about these two? This is Eric Kandel and Brenda Milner, of course, who are way over 90 years old and still very sharp. And that's what we would now call the super agers. So I think we can learn a lot from these types, people who age extraordinarily well without the slightest cognitive decline. And that might be equally relevant to understand neuropathology than studying Alzheimer's disease or Parkinson's disease. So what prevents them to get the disease? And this work also by Dr. Hönig, who found that not only do the super agers have no amyloid pathology, of course not, but also they don't have the slightest degree of tau pathology in their brain. So a little bit of tau pathology seems to be quite normal with aging. There's a term called age-related tauopathy, or primary age-related tauopathy. And it's hotly debated, of course, but there seems to be a little bit of tau pathology building up in the parahippocampal area and hippocampal area in most of us. But the super agers seem to be even clearer of that. So I think we want to learn more about what really drives that effect. We also want to be super agers. And with that, I'd like to leave you and thank my group of super agers, hopefully in the future. Yeah, thank you all. Thanks for the invitation, and hope to see you soon in person somewhere. So we have a couple of minutes left to punch in some questions.
Okay, so thank you, Professor Eimeren, for a nice overview. I think it resonated a lot. It was said before on a smaller scale to put these things in a broader perspective. So
thank you.
Great
job with the conference, by the way.
Yeah, I see we have, since we have a couple of minutes until the 7, I see we have one question currently, and I hope more will follow. So the first question is there any lifestyle characteristic that resemble the role of education, but in motor domains?
Yeah.
So perhaps sporting activity, or some professional athletes suffer less from Parkinson's disease.
Well, it depends on what kind of sports you do, probably. So we know that there could be an association with contact sports developing Parkinson's disease, such as boxing, for example, or other contact sports, and similar diseases. But it's highly likely that physical activity, and that doesn't always mean sports. It could just be that you're always out and about in nature and doing the gardening and all that. That could help and be a protective effect. We don't know that for sure. So we don't know the drivers of motor reserve. We think it might be that factor, but it could also be that motor reserve isn't really about the motor system at all. We know that the Parkinson's, we think of Parkinson's disease as a movement disorder, of course, it's a motor disease, but it's not really affecting primarily the motor system, right? So it's not like it's a palsy, it's called shaky palsy in the beginning, but it's more, it could be more about the planning and the motivation of movement, and therefore the factors that might promote the reserve mechanism or the resilience to the pathology might also be educational, might also be motivational. So if you're a highly motivated person, but that also could, tie in with the sports. If you're able to motivate yourself to do a lot of physical activities, that might help. So that's exactly the thing that we want to find out with this study, the DeMoco study. What is really the driver of motor reserve?
Okay, thank you. And I think we can tackle one more question. It's about the Super Agers study that you have mentioned. How large of a sample size were you looking at?
Well, that's a good question. I don't have the exact numbers, and obviously it's a limited data set. It's from the ATNI cohort. So the way that Merle did that, and I will have to ask her again about the exact numbers. The way she did that was you take the healthy control data set and then, of course, look at an old life over 80, basically, and compare the cognitive scores of them. And if they score better than the majority or the average of the elderly control group, then they are deemed super agers. That's a very pragmatic approach to that. And I think we need to, and that's of course not a study that was engineered to look at that. So I think we need really studies, population-based studies, much more looking in that direction of healthy agers and super agers. So it's probably in the realm of a couple of dozens. So 20, maybe 30, something like that.
Okay.
Not enough to really be very confident about it.
Yeah, but we hope that we will see more and more super agers in the future, right?
Yes, that's the hope.
And maybe one final question. Do you think that these resilience factors would explain the heterogeneity in the clinical presentation of various neurodegenerative diseases?
Yeah, that's I strongly believe that, because it's so striking. If you I mean, I'm involved in the nuclear medicine department, so I do also see the DaT SPECT images. So I also part of the team that looks at the PET images and the SPECT images. But I also have my own outpatient clinics for Parkinson's, and I refer all of to establish the diagnosis of Parkinson's disease, we always do a DaT SPECT, because there are so many surprises. You think that this is Parkinson's disease, and then it turns out it's not, or you think it's essential tremor, and it turns out it's PD. So that can happen. So we think it's a needed approach. And then in some patients, you're amazed by the degree of pathology that you can also already see, even though there are only little symptoms. And in other patients, it's the opposite. So I think that this is really something so this kind of resilience towards pathology is really very important and under studied still.
Okay, thank you. And the last question is regarding the asymmetry in neurodegenerative disorders, I mean, especially in Parkinson's disease. Do you have any insight what would be the cause of this asymmetrical presentation and brain pathology?
It's very funny that you asked me that, because that's my go-to question if I have a very great speaker like Tony Lang or whatever, not me, but someone who really knows their stuff. And I don't have a good answer to that. So it's something that intrigues me for a very long time. It doesn't make sense. It really doesn't make sense that PD is asymmetric. It if you think about it. Why should it be? It doesn't make sense. So, but it's the case, so we have to deal with that. And it could be, and that's something that I recently thought of more with the evidence of this mismatch between Lewy body pathology and the loss of dopamine neurons. Maybe the Lewy, and we don't know if the Lewy body pathology is asymmetric, by the way, because nobody looks at that. Now people start to look at that, at both hemispheres in the post-mortem, but they tend to look only at one hemisphere, right? That's what pathologists do. So they need to change that habit to really explore if the Lewy body pathology is really asymmetric or not, and if that correlates with the asymmetric presentation of PD. That's clearly the case in most patients. And maybe the Lewy body pathology isn't asymmetric, and the FDG pattern, for example, and Maria mentioned that it's mostly symmetric, right? So there are some asymmetries there, but it's mostly symmetric, and that's interesting because Parkinson's disease is asymmetric. So why should it be symmetric? So maybe PD isn't asymmetric. Maybe only the dopamine system is asymmetrically affected by PD, right?
Yeah. Okay, and I think that now we ran out of time. So I thank you once again for the discussion and for the plenary talk.
Sure. Thank you. Bye.
Bye, Professor Eimeren. And just a few last words for the audience. The program is following tomorrow, starting at 11 with the poster session, and you can already see the posters on our website. If you scroll all the way down to the website, you will see the short oral presentation section. And you're welcome to look through those and answer any burning questions that you might have, and they will be answered tomorrow during the poster session. Furthermore, you're all welcome to join us tomorrow when we have an interesting plenary lecture symposium, in parallel running also the clinical neurophysiological symposium, and then a workshop on proteinopathies. And finally, in the evening, we have the Neuroscience and Society dialogue with an interesting speaker. One last thing before we say goodbye, the CME poll for the continuing medical education credits will be still running for a few more minutes on the website, so you can still register if you haven't done yet. And we can also see the cloud graph of people joining us from all over the world today. I think there were a couple of hundreds joining throughout the program, so gather in at least in equal amount tomorrow. And goodbye.